So we've been talking about crosses, specifically Punnett squares that go through and try to cross and figure out probabilities of how these things work. This is a way oversimplified version of how genetics works, which is actually not my favorite thing to talk about, considering that it really doesn't happen like this a lot, because there's a lot of complicated things that go on. A dihybrid cross is a very commonplace example of saying there's two heterozygotes crossing. But there's other ways to do this. If you have a homozygous dominant parent and you have a homozygous recessive mother, father, whatever, one parent is blue, one parent is red, these are their genes, right? They're homozygous, which means that this parent has two copies of this book that are red, and this parent has two copies of this book that are blue. And considering they can only give off one of those books after they are done mating, in looking at it, this square represents one child that might get that. And for this one, every single square is going to look like this first square. Red's going to come across, blue's going to come down. Red's going to come across, blue's going to come down. Red's going to come across, blue's going to come down. Blue's going to go down, red's going to come across. The entire thing is going to be a heterozygote. 100% heterozygotes. When you have a homozygous dominant and a homozygous recessive parent, everything's going to be heterozygotes, which is actually how inbreeding works for Gregor Mendel to figure out how to get dihybrid crosses that we covered yesterday to be able to figure out the 75 to 25% ratio. Inbreeding them over and over again to actually get homozygous recessive and homozygous dominant so that we could do the pea plants and figure out the percentages was how we did all of this. That is how you get two parents that are homozygous dominant, homozygous recessive to have all children that look dominant because they'll all have the blue allele showing through.